At a quick glance, the life of a cell, its cell cycle, is pretty routine. It's either actively dividing or preparing to divide into two daughter cells. The cell cycle itself has an interphase, made up of subphases G1, S, and G2, during which the cell is preparing for division, and mitosis, during which the cell actually divides. During the S phase, the cell performs DNA replication, which is when its 46 chromosomes are duplicated so that each daughter cell can have its own copy of the genetic material. A single chromosome is made up of a single DNA molecule that has two strands, which wrap around each other to form a double helix. Each single strand of DNA is composed of a sequence of 4 types of nucleotides, which are the individual letters or building blocks of DNA. Nucleotides of DNA are made up of a sugar, which is deoxyribose, a phosphate, and one of 4 nucleobases, adenine, cytosine, guanine, and thymine, or commonly A, C, G, and T for short. The nucleotides on one strand form hydrogen bonds to complementary nucleotides on the other strand. Specifically, A bonds with T via 2 hydrogen bonds, and C bonds with G via 3 hydrogen bonds. Additionally, the 2 DNA strands also have a direction, meaning one of them runs from the 3' end to the 5' end, while the other one runs from the 5' end to the 3' end. Kind of like 2 snakes coiled up together, but facing in different directions. DNA replication can be described as semi-conservative. That means that each strand of the double helix acts as a template, based on which new, complementary strand will form. Eventually, the original chromosome will split into 2 exact copies, each made of one of the original strands and one of the newly made ones. Overall, DNA replication has 3 steps: initiation, elongation, and termination. Initiation kicks off when a group of proteins get together to form the pre-replication complex. This pre-replication complex looks for specific nucleotide sequences along the DNA strand, called origins of replication. And yes, we're talking plural. That's because our DNA strand is so long that DNA replication actually starts in several origins along the chromosome simultaneously. These special nucleotide sequences have a ton of A and T bases. Because A and T are joined by only 2 hydrogen bonds, the enzyme DNA helicase has a relatively easy time separating the 2 strands. This creates a replication fork, with the 2 prongs of the fork being the 2 strands that are separated from each other. Single strands of DNA can get a bit unstable, so to help keep them from getting back together again, helper molecules called single-stranded DNA binding proteins come in and bind to each of the lonely strands. Also, as DNA helicase breaks down bonds, the segments of DNA ahead of it start to overwind, meaning the double helix becomes more tightly wound. You can try this with a close friend who has braided hair. If you pull the braid apart in the middle, then the ends get tighter. But make sure it's a friend and not a stranger. Overwinding of the DNA can slow down replication. So the enzyme DNA topoisomerase works ahead of the DNA helicase to loosen up the tight DNA coils. It achieves this by gently snapping one strand, loosening the overwinding tension, and then patching it back up, tension-free. The second step is elongation, and for that we need a new enzyme, RNA primase. Yep, you heard that right, RNA this time. This is kind of like DNA, but it uses ribose instead of deoxyribose in its nucleotides. And instead of thymine, it uses uracil. So what RNA primase does is hover around and randomly synthesize small lengths of RNA which are only a few nucleotides long, called RNA primers. Just by chance, at least one of those RNA primers will be complementary to the open DNA strand, and it'll bind. The enzyme DNA polymerase can then latch onto this short double-stranded segment, and from there the DNA polymerase can add more complementary nucleotides to the template strand. Now, the DNA polymerase moves in a 3' to 5' direction along the template strand, since only the 3' end offers the OH group required for synthesis. The complementary strand is formed in the opposite direction, 5' to 3'. As a result, replicating this DNA template strand is smooth sailing, and this new strand of DNA that forms is called the leading strand. Things are a bit more complicated for the other DNA template strand, that runs from the 5' to 3'. That's because the RNA primer that's anti-parallel is facing the other way. For this new strand of DNA to grow toward the replication fork, DNA polymerase would have to add nucleotides to the 5' end of the primer, but that's not possible. To solve this issue, RNA primase lays down a number of primers that bind to different spots along the length of the DNA template strand. This offers a lot of free 3' ends. DNA polymerase can then add nucleotides to the 3' ends of each of these primers. We call these growing fragments of DNA Okazaki fragments. And they each grow away from the replication fork until they meet up. Unfortunately, DNA polymerase cannot join two Okazaki fragments, so they're joined together by another enzyme, DNA ligase, resulting in a finished strand of DNA called the lagging strand. Now, the final step of DNA replication is termination. At the end of the chromosome is a region of DNA where there are repeating sequences of TTA-GGG nucleotides. This part of a chromosome is called the telomere. The DNA sequence in the telomere signals the DNA polymerase to hop off of the strand right before replicating the DNA at the very end. Fortunately, the repeating TTA-GGG sequences don't code for anything, so no genes miss out on getting replicated. The telomeres are like those plastic bits on the shoelaces that only exist to protect the shoelace itself. And the result is that each time the DNA strand is replicated, the new strand is just slightly shorter than the template strand because a bit of the telomere itself is lost. That, however, means that we have a strictly limited amount of possible divisions available to our cells before those divisions actually start shortening our genes, which, as you might imagine, is bad. This limit is known as the Hayflick limit. Alright, as a quick recap. DNA replication happens in 3 steps: initiation, elongation, and termination. Initiation starts with DNA helicase and topoisomerase helping to unwind the DNA. During elongation, RNA primase creates RNA primers, and DNA polymerase creates a leading strand through a single primer, or a lagging strand through creation of Okazaki fragments that eventually merge together. Finally, during termination, the 2 double strands, each made out of one new and one old strand, separate, slightly shortened at the telomere region.